hope everyone's having a good day. I'm going to give a presentation on one aspect of my PhD dissertation project, controlling response time in optochemical CO2 sensors via crosslink density of PDMS membranes. My name is Teshaun Francis. I'm a PhD candidate. My advisor is Dr. Lin. And what we are interested in is respiratory disease. Now, the respiratory control system starts in the lungs. The oxygen travels to the tissues via the cardiovasculature, and we have chemoreceptors, mainly carbon dioxide chemoreceptors, which detect the carbon dioxide, which is a byproduct of respiration. That is the chemoreceptors feed into the ventilatory control center in the brain, and then that tells the lungs how hard to breathe and how fast to breathe. Now, basically, respiratory disease affects this this figure illustrates where respiratory disease affects that control loop. Ventilatory dysfunction can exist in the lungs, can exist in the cardiovasculature, or can exist in the brain or in the nervous system. So, a solution that we're aiming towards is a artificial ventilator with a closed loop adaptation. Um, this is the long-term goal, but basically what we need is a carbon dioxide sensor to bypass the chemoreceptors in the body, in the body because we can't really access them. So we have artificial ventilators, mechanical ventilators, diaphragm pacing, but we don't have the CO2 sensor to get the feedback. So what we want to do is develop a CO2 sensor system to provide feedback for a ventilator and then create that closed loop adaptive system. So where we are now is we're developing an optochemical CO2 sensor, which the opto means optical, the chemical is chemical. So it uses the mechanism is called the Severing-House mechanism, and we transduce carbon dioxide concentration into an optical signal via a sensing media. And the sensing media right now is aqueous, so we have to encapsulate it in a membrane. And the membrane is provides some structural. It provides a structural component to the sensor, but it also allows for gas separation. So this keeps the sensing media separated from the external environment, but the CO2 can pass through it. Now, what we've found is these chemical sensors are based on multiple factors, diffusion and kinetics, but overall, we're finding that the response time is poor. And so, as part of the research, we were interested in improving the response time, and there are many different ways to do that. And I think the most obvious one was, well, the membrane is literally in the way of the CO2 in the external environment and the CO2 and the sensing in the sensing media. So we focused in on the membrane diffusivity, and this can just be described by its fixed second law of diffusion. So the flux through the membrane is proportional to the diffusion coefficient of the membrane, the and then the second derivative of the CO2 the CO2 gradient divided by the second derivative of the distance, or the thickness of the membrane, or the distance. So therefore, there's like the thickness of the membrane. So we were interested in. I'm not doing that, by the way. I don't know why the slide's moving forward, but we were interested in either, well, we can't control the CO2 gradient that is controlled by the fluctuation in the environment, but we can control the membrane thickness. However, we don't want the membrane to be too thin or else it'll be fragile and might puncture or break or something. So after minimizing the thickness, we were interested in maybe, we were curious, maybe can we target the diffusion coefficient? And so that's where we were interested in improving the response time by improving the diffusivity by somehow modifying the diffusion coefficient. So we took like a deep dive into what controls diffusion coefficient, and it's kind of complicated, but on a basic level, PDMS is what we're using, polydimethyl siloxane. It's a cross-linked polymer, which means that there are pre-polymer chains, so they're like strings, and then you have this curing agent, which which links the chains together. So that's what the polymer is literally one solid piece these chains connected these chains connected by other chains. So you get this cross-linking. And very important factor of cross-linking is the cross-link density, the number of cross-links per unit volume. And so, well, well, maybe this is something we can study. We did some research into cross-link density and found that cross-link density increases free volume in polymers, which improves diffusivity. Okay, so it generally improves diffusivity. PDMS with reduced cross-link density has higher diffusivity to oxygen gas, which showed a specific application that showed that gas diffusivity of not carbon dioxide, but it was oxygen. A gas diffusivity was improved by modifying the cross-link density. And so um, the last thing we found is the cross-link density is literally just controlled by the number or the curing ratio to prepolymer mix ratio. And that's something you control when you fabricate the PDMS. And that's what we were doing anyway. So we we basically look at this equation. The carbon dioxide rate over time is equal to this. It's equal to the diffusion, but also equal to kinetics. But if we just focus on the diffusion part, we can directly modify D in a way that we may not be able to quantify right now, but we can change it through the curing ratio. So we came up with an experiment. We have prepolymer mix, we have curing agent, we have the process for making CO2 sensors. Can we just change the curing ratio to two different levels and then see how that affects the response time, changing nothing else about the sensors? So we have um, yeah, and then that's basically our hypothesis. We have two curing ratio levels, which is 1 to 20 and 1 to 5. We're calling the 1 to 20 low and the high is 1 to 5. So we make the sensors through, I'll just tell you the process, right? We start with, well, it's two kind of separate processes, making the sensing media and then making the PDMS membrane. The sensing media is in the bottom left corner. It's made of deionized water, HPTS, or pyranine, which is called the pH indicator. That's part of the Severing-House mechanism, the CO2 sensing mechanism, which we could talk about if you want to, but just know it's an important part. And then there's a buffer, which is actually just baking soda. And we create the sensing media, and then once that's done, we fabricate the PDMS membranes using the prepolymer mixture and the curing agent. Then we use a mold to cast the mixture, and that gives us PDMS sheets, which we can use to assemble a CO2 sensor, which is a picture of one is on the right. So they're fairly small, but they're not. They're fairly small, and the idea is that they are flat and um, they're flat and lens-like is the best way to describe it. It's like a lens. So in this way, we can do some very simple absorption spectroscopy measurements to um, monitor the absorbance through the sensors. So this is what our experimental test bed looks like. We have a collimated light source transmitting, creating a transmission beam through the sensor, which is in a test chamber, and we capture the transmission through the sensor with one spectrometer, and we capture a reference signal with another spectrometer. And then we do some signal processing to calculate an optical signal, which we are monitoring in real time. At the same time, we have a pump, a room air pump, and a canister of carbon dioxide, and then we have control valves to create a gas mixtures of different levels. and then we can create CO2 step functions, which we are monitoring with another CO2 sensor, which is just measuring the partial pressure of CO2 with it's using infrared light. So moving quickly. Yes, so the basically we found that the curing ratio had a significant effect on the response time. It was very apparent. Using the low ratio, we had much lower response time than the high ratio, but we also found something else that was interesting that we didn't see before. In the high curing ratio group, there was a huge discrepancy. There's huge discrepancy between the forward and the reverse response, and it appeared to us that that making the sensors slower, at least in this case, seemed to exaggerate this difference between the forward and the reverse. So we came to several conclusions. The most obvious and useful one being that while they're still kind of slow, we're still working on making the sensors much faster. The low curing ratio group had a much faster response time, so that was apparent and immediately useful for us. But also, we had more questions. Where's this asymmetry coming from? And is it an effect of the curing ratio or is it intrinsic to the sensor? So we want to do some setting of the analytical solution to see if we can um, differentiate between the different rates, the diffusion rates and the kinetic rates, and see which one is contributing to that asymmetry. We also found that, just to be clear, this is the last thing. Um, Changing the curing ratio doesn't change the density of the PDMS. We just confirmed that just to make sure that there wasn't less material overall. It's actually the same density, but there was a significant change in mechanical stiffness. And we found that was true, and we found some other papers which confirmed that this is normal. So basically, um, yes, we can make the sensors faster with very little change to our fabrication process, but then we are indirectly making them more flexible, which flexibility is not a big issue, but maybe fragility is. We're wondering if we can extract the diffusion coefficient of these two levels of curing ratio. We're also interested in curing ratio effects on hydrophobicity and the interaction effects with curing temperature. These are all just things that we're working on. But yeah, in conclusion, change the curing ratio, change the response time in CO2 sensors, but also more elastic. Is it more fragile? That's a trade-off we might have to consider. I'd like to acknowledge, the department, funding sources, and of course my advisor. I'll take